we presented results of our randomized phase 2 trial of SWOG 1505. We compared two perioperative chemotherapy regimens, modified FOLFIRINOX and nab-paclitaxel, which are the standard frontline regimens for metastatic disease. This was the first study to test them in the perioperative setting for resectable pancreas adenocarcinoma. And we found that of the 147 patients we enrolled, with 102 eligible, there was a 29% proportion of patients who were found to be ineligible on central radiology review. Of the 102 eligible patients, 96% in each arm approximately started chemotherapy, 85% completed all chemotherapy, and 73 and 70% reached surgical resection in the FOLFIRINOX and nab-paclitaxel arms, respectively. The R0 resection rate among those who got resected was 85%, and the primary endpoint of 2-year overall survival did not reach its pre-specified threshold. The median overall survival in each arm was about 23 months, and it was not remarkably different between the two arms. So the take-home point was that the study did not meet its pre-specified threshold of overall survival improvement, but we had important lessons that we learned moving forward. It was a randomized phase 2 trial. We treated patients with resectable pancreatic adenocarcinoma. They were enrolled and randomized to one of two arms. The arm 1 was modified FOLFIRINOX, and arm 2 was gemcitabine nab-paclitaxel. In each arm, patients received 3 months of chemotherapy and then underwent restaging scans. After those restaging scans, if there was no disease progression, they underwent surgical resection. And after surgical resection, they then underwent 3 more months of adjuvant chemotherapy with the same regimen that they were assigned to at the beginning. So the number one lesson is that perioperative chemotherapy is safe and feasible. Patients, 85% of them completed preoperative chemotherapy, and 73 and 70% underwent resection. So we established safety. We also established metrics in this setting. The adjuvant chemotherapy trials for resectable pancreas cancer enroll patients after resection, and a lot of patients don't even make it to that point. But in our study, this was all comers. Patients enrolled from day 1 of pancreas cancer, and we established metrics of outcomes in terms of overall survival, disease-free survival, and R0 resection with these modern chemotherapy regimens. And the third very important lesson we learned is that 29% of patients, for example, in our study had more than eligible disease for resectability criteria, and therefore moving forward, there has to be a lot of importance placed on quality control to ensure that we are indeed enrolling resectable disease in these studies. Now we are doing correlative studies to identifying sarcopenia on baseline scans to assess whether that might be related to ultimate outcomes, as well as detailed immunologic and immunohistochemical evaluations of surgical specimens to see if we might find predictors of response in terms of immune infiltrates into the tumor, as well as pathologic complete response as it might relate to overall survival outcomes.